This chapter, we're going to cover cells, the living units. So, when we look at the concepts as we progress through this chapter, we need to remember that the cell is the smallest living unit. And whatever its form or however it behaves, the cell is the microscopic package that contains all the parts necessary to survive in an ever changing world. It follows then that the loss of homeostasis in cells underlies virtually every disease. So, I have a brief video for an introduction to this chapter, and it comes from Mr. B's Anatomy, which you can subscribe to him on YouTube. What he does is he has a lot of his students come in and recap a unit. And so, this one is one of his students recapping cells, the living unit. Don't forget to read your chapter and complete your assignments for the week.
Cells, the living units. The cell membrane is what makes up the outside of a cell. It protects the cell from the outside environment and controls what can leave and enter the cell. This is a property that we call selectivity permeability, meaning some things can enter the cell while others cannot. If you take a closer look, this membrane structure is also known as a fluid mosaic model, which is made up of lipids, proteins, and carbohydrates. We are going to be zooming in on a plasma membrane wall-like structure, which has a phospholipid bilayer that is made up of two layers, the hydrophilic heads on the outside, which are water-loving, and hydrophobic tails on the inside, which are afraid of water. Next, you have your glycolipids, which act as a surface receptors that stabilize the membrane and serve as cell surface markers placed on top of the polar heads of the phospholipid molecule. Also scattered in the lipid bilayer are cholesterol molecules, which help to keep the membrane fluid consistent. Membrane proteins do not interact with hydrophobic core of the phospholipid bilayer. Instead, they are usually bound to the membrane indirectly by interactions with integral membrane proteins or directly by interactions with lipid polar head groups. Proteins do most of the work in cells and are required for the structure, function, and regulation of the body's tissues and organs. A good way to remember all these functions is the word creation. This stands for cell-to-cell recognition, receptors, enzymes, attachment, transport, and intercellular adhesion. Let's take a closer look at these. This is cell-to-cell recognition. Cell-to-cell recognition is one of the ways by which cells communicate with one another. Then there are receptors, which carry cell messages inside the cell. Enzymes catalyze or cause reactions inside or outside of the cell. Attachment proteins bind other proteins to different cell structures. Transport proteins serve the function of moving other materials within an organism. Last but not least, we have intercellular adhesion, which helps cells stick together. Membrane carbohydrates are usually attached to the outer surface of the cell membrane. They act as antennas and are used by the cells to identify each other. There are two types depending on what they are attached to. Glycolipid, a carbohydrate attached to a lipid, and glycoprotein, a carbohydrate attached to a protein, as shown in the picture. Now let's take a look at specializations of the plasma membrane. Microvilla are finger-like extensions that increase the surface area of cells and minimize any increase in volume. They are involved in a wide variety of functions, including absorption, secretion, and cellular adhesion. Another specialization are junctions. There are two types of junctions. The membrane junctions, these are structures that consist of multi-protein complexes that provide contact between neighboring cells or between a cell and an extracellular matrix. Tight junctions form waterproof connections between cells, like little separate compartments. Junctions are holes and passageways between cells that allow for intercellular communication, where ions and molecules can flow from one cell to another. The final specialization is decimones. Decimones are extra strong anchoring junctions that act like a Velcro. These give very strong bonds to give mechanical strength to tissues. You may be asking yourself, how do materials move across the plasma membrane? Well, let's take a look. The plasma membrane has a selective permeability. This means that it allows certain molecules or ions to pass through it by means of active or passive transport. Now, you may be asking yourself, what is active and passive transport? Passive processes include simple diffusion, facilitated diffusion, osmosis, and filtration. Active processes include active transport and vesicular transport. Let's take a closer look. Simple diffusion is a process where a substance passes through the membrane without an aid in the membrane protein. Facilitated diffusion carries proteins and channels to pass through a membrane. Osmosis is the diffusion of water across a semi-permeable membrane. This can cause pressure. Two types of pressure are hydrostatic and osmotic. Hydrostatic pressure is a pushing on the liquid with a pump or gravity, for example, blood being pumped by the heart. Osmotic pressure occurs when a solute is being dissolved in a solution. When there is an unequal solute in the solution, pressure forms. Finally, we have to look at toxicity and osmosis. There are three types of toxicity: isotonic, hypertonic, and hypotonic. Imagine that you're in a hospital and the nurse hooks you up to an IV. What's going on in that IV? Is it just water? No way. If you were pumped full of water, your blood cells would just burst. That IV is full of saline, a liquid with the same concentration of solutes as your blood cells. This is an example of isotonic solution, when two solutions separated by a semi-permeable membrane have equal concentration of solutes and water. A hypertonic solution is one where the concentration of solutes is greater outside the cell than it is inside. If so, your cells would shrink. Finally, a hypotonic solution is one in which the concentration of solutes is greater inside the cell than it is outside. This would cause your cells to burst. Filtration plays an important role in movement. It is the movement of water and solute molecules across the cell membrane due to pressure. There are two active processes: active transport and vesicule. Active transport is the movement of ions or molecules across a cell membrane into the region of higher concentration, assisted by enzymes and requiring energy. Vesicle transport is responsible for molecular traffic between varieties of specific membrane-enclosed compartments and consists of exocytosis and endocytosis. Exocytosis is where materials are exported out of the cell via secretory vesicles. Endocytosis is the process of capturing a substance or particle from outside the cell by engulfing it with the cell membrane. There are two types of endocytosis: phagocytosis and pinocytosis. We can remember phagocytosis by thinking of it as a Pac-Man. Stands for cellular eating. It is the process by which certain living cells, called phagocytes, ingest other cells or particles. Pinocytosis is the ingestion of liquid into a cell by the budding of small vesicles from the cell membrane. Cytoplasm is the jelly-like material that makes up much of a cell inside the cell. Inside the cytoplasm is the cytosol and many cytoplasmic organelles. Cytosol is the liquid found inside of cells. There are 9 different cytoplasmic organelles that you should know of: mitochondria, ribosomes, endoplasmic reticulum, rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus, lysosomes, peroxisomes, and cytoskeleton. Mitochondria produces ATP and energy. Ribosomes make protein. There are 2 types of ribosomes: free ribosomes, and membrane-bound ribosomes. The rough endoplasmic reticulum has ribosomes and produces proteins. The smooth endoplasmic reticulum produces lipids. The Golgi apparatus packages, modifies, and transports the cell. We can remember this by thinking of FedEx. Lysosomes remove waste from the cell, and peroxisomes are used to break down fatty acid chains. The cytoskeleton forms a framework for the movement of organelles across the cytoplasm. It is made up of tiny rods and filaments called microtubules, microfilaments, and actin filaments. Finally, we have centrosomes, centrioles, cilia, and flagella. Centrosomes are where centrioles are found. Centrioles are only found in animal cells. Cilia and flagella move liquid past the surface of the cell. Cilia move or sweep mucus out, and flagella is a whip-like appendage that allows many protozoa, bacteria, and sperm to swim. There are three types of inconclusions or storage areas: glycogen, which is sugar, lipids, which are fat, and melanin, which gives our skin tones. Now let's talk about the control center of the cell, the nucleus. The nucleus houses DNA and it is enclosed by a membrane. We can break the membrane down into four important parts you need to know: nuclear envelope, nucleoplasm, nuclei, and chromatin. Covering the nucleus, we have the nuclear envelope, which is composed of two phospholipid bilayers that controls what comes in and out. We can kind of think of this as the bodyguard of the nucleus. Inside the nucleus, you will find the nucleoplasm. Think of this as the swimming pool for nuclei and chromatin. The nucleoplasm is a thick fluid that surrounds these two structures, and it is important because many substances, including enzymes, are dissolved here. Finally, we have chromatin, the active form of DNA. The function of chromatin is to package DNA into a small volume to fit into the nucleus of a cell and protect the DNA structure and its sequence. Ever wonder what the life of a cell looks like? Wonder no longer. Here is the cell life cycle. The cell's life cycle can be broken into two phases: the interphase, the mitotic or cell division phase. Looks like an interphase. Interphase is the period from cell formation to cell division. We can break down interphase to three steps: G1, S, and G2. During G1, cell grows and synthesizes proteins needed for mitosis. S phase follows G1. Its major function involves replicating the cell's DNA. The S stands for synthesize. By using enzymes, the cell copies the DNA molecules of each chromosome. Finally, after S phase, the cell enters G2 and it involves another period of growth. The cell synthesizes more proteins, organelles like mitochondria, to grow and divide. This stage prepares the cell for division. The mitotic phase comes next and is broken down into four stages: photophase, metaphase, anaphase, and finally telophase. During the photophase, the nucleus disappears, mitotic spindles start to form, and DNA condenses into chromosomes. Next is metaphase, where chromosomes line up at the line of the cell to form the metaphase plate. The chromads of each chromosome are attached to a microtubule. During anaphase, the paired chromosomes separate and move towards the opposite sides of the cell, causing them to move further apart from each other. By the end of each cell cycle, they should have a complete set of chromosomes. Telophase is the opposite of photophase. The new nuclear envelope will form around each set of chromosomes and the nucleus reappears. This completes mitosis. The final step we need to discuss is cytokinesis. Cytokinesis is not a phase, but a separate process in itself. A cleavage furrow develops where the metaphase place used to be pinching off the separated nuclei. This is where the cell divides. The last topic we are going to discuss is protein synthesis. Protein synthesis is one of the most important fundamental biological process by which individual cells build their specific proteins. To understand the role of RNA, we first need to understand how it differs from DNA. RNA is single-stranded, and it has a sugar ribose instead of a deoxyribose. Three forms of RNA work together to carry out DNA's instructions. RNA can be thought of as the actual machinery that makes proteins. RNA has three forms: mRNA, tRNA, and rRNA. mRNA, also known as messenger RNA, carries the genetic info copied from DNA in the form of a series of three base code words, each of which specifies a particular amino acid. We can think of this as the recipe for the protein. tRNA, other known as transfer RNA, decides what amino acid is needed by looking at mRNA. Once it recognizes the particular amino acid it's needed, tRNA moves that amino acid into a chain of amino acids. Finally, rRNA, or ribosomal RNA, associates with a set of proteins to form ribosomes and assemble amino acids into protein chains. There are two stages associated with protein synthesis: transcription and relation. Transcription takes the information encoded in DNA and encodes it into mRNA. This happens in three steps: initiation, elongation, and termination. RNA polymerases pull apart the strands of DNA, double helix, so the transcription can begin. RNA polymerase elongation the mRNA strand one base at a time and unwinds DNA helix in front and behind of it. Finally, when the polymerase reaches a special base sequence, transcription ends and the newly formed mRNA separates from DNA. Translation is where information carried by mRNA is decoded and used to assemble proteins. In conclusion, you now should know all about the cells after completing this video: what they are made out of, how they move, and the cycle in which they live their lives. Thank you.